This is part 2 of microbial nutrition, and we're going to talk about gases. The first gas we'll talk about is oxygen. There are several categories that you can see here of oxygen use in bacteria, and what is shown here is a special medium that is used primarily to figure out their oxygen use, and these are called thioglycolate tubes. So thioglycolate is the material in these tubes, is the medium. And when you put a bacteria in this tube, they tend to grow within the oxygen content that they require. So the thioglycolate basically is a tube, and then as you would imagine, the oxygen concentration is the highest at the top, where it meets the oxygen in the tube in the atmosphere, and then as you go down to the bottom of the tube, the oxygen levels will drop, and so things that grow at the bottom of the tube will basically have like very little oxygen or no oxygen present. So let's look at what our choices are, right? So we have a category, it's called obligate aerobes, and these are microbes that require oxygen and will die if oxygen is not present. So the growth of these organisms in this kind of medium will only be at the top where there is an ample amount of oxygen. Then the opposite, obligate anaerobes, will die in the presence of oxygen. So this is where they would grow. They would grow at the very bottom of this tube. Then we have the facultative anaerobes, and facultative anaerobes can do both. They can grow with oxygen, right? We looked at that with cellular respiration. They make ATP way better than they with oxygen than if they were to ferment without oxygen. So you would find that with oxygen, they grow much better because they can just efficiently make a lot of ATP and you get more robust growth. However, if you see the low oxygen area, they still grow, right? Not as well, or not as like as much as the oxygen region, but they can still grow because they can ferment, right? Remember, they can ferment without oxygen. So you'll find that these tubes heavily at top, but then they're turbid all the way through. Then we have something called aerotolerant anaerobes. So these are guys that are pretty much evenly distributed in the test tube, that their oxygen content, they can grow in any of these conditions. So they're basically anaerobes, but they can tolerate oxygen. So they're not like killed by oxygen. So in this one, the obligate anaerobes, these guys are killed by oxygen. They cannot handle oxygen because oxygen does make some nasty reactive oxygen species that can harm them. And then we have something called microaerophiles. So these guys really like just a little bit of oxygen. So not too much, right? The top is going to be too much oxygen for them. They like this sweet spot where it's like in the, there's oxygen present, but not a lot. So those are called microaerophiles. This is an example of the real test. So you can see test tube 1, we have heavy growth at the top. We have a pellicle, so that's an aerobe. Then we have something that's heavier at the top, but we can see turbidity throughout. So this is going to be our facultative anaerobe. We have aerotolerant, which is pretty much even all the way through. And then we have an obligate anaerobe. This is kind of a little bit different than the picture we saw, but we can see a very clear area where there's a high oxygen concentration and there's zero growth. Only when you have a lower oxygen content do we have growth. So that's going to be our obligate anaerobe because it cannot grow at all where there's oxygen. A way that we can grow anaerobes in the lab, right, because there's air all around us, is to trap them in a jar that is devoid of oxygen. So there's a special thing called an anaerobic jar, and there's a chemical way to remove the oxygen in there. This is something we'll do in lab as well, so we'll just wait on the details for that. Other times, if you work at a fancy lab, they'll have an anaerobic chamber, and you can just put these gloves on so nothing needs to open the chamber, and you work with the bacteria in there. This is something called gas gangrene. This is something that is caused by an anaerobe called Clostridium perfringens. We will learn about this guy later. So when you have an anaerobic infection, these bacteria cannot grow with oxygen, right? Your blood is full of oxygen. Your blood is carrying oxygen on hemoglobin, right? So this kind of infection happens when you have an injury, when you have some kind of issue with the tissue, where the blood supply is blocked so that you're creating an anaerobic environment. And then once you have an anaerobic environment and you have Clostridium perfringens in this area, this is not a normal flora microbe, by the way. This is a guy that invades our body. But if you have this condition where the blood flow has been blocked by some injury, then the bacteria can grow. And the gas comes from the fact that the bacteria are fermenting, and gas is a product of fermentation. So you'll find that tissues will have these really big areas of gas bubbles that are created from the fermentation. And gas gangrene is really awful because it can cause tissue necrosis, which is dead tissue. So this is a diabetic foot ulcer where in these foot ulcers, the tissue has been damaged a lot from diabetes, so that it's a perfect place for Clostridium perfringens to grow and cause gas gangrene. Here is why oxygen can be toxic to a lot of microbes. Because oxygen is a really reactive molecule, O2 is reactive, it can create something called a singlet, a superoxide radical, and a peroxide anion. These 1, 2, 3 are all really toxic to the cells. They can destroy sort of like their normal molecules. They can, destroy DNA. So they're basically really, I was like toxic. So all of these forms, as you can see, contain oxygen. And we breathe oxygen all the time, right? So aerobes, obligate aerobes, use oxygen, and we're making these all the time. Oxygen is going to do this no matter what. It's going to make singlets, it's going to make superoxide radicals, to make peroxide ions. So because we evolved using oxygen in the electron transport chain, we have ways to basically neutralize these radicals. So enzymes, superoxide dismutase is an enzyme. Catalase and peroxidase are enzymes that our cells make just for the prevention of the buildup of these toxic forms of oxygen. These toxic forms of oxygen are called reactive oxygen species, or ROS. So to clear and destroy the ROSs, these enzymes will break them down. One in particular is catalase, which we'll look at in class, in lab, and it converts, this is hydrogen peroxide. Okay, so we know that hydrogen peroxide is used to kill bacteria. We put it on our cuts and wounds, right, to clean them, to hopefully kill bacteria. So a lot of H2O2 is very deadly to cells. When you make hydrogen peroxide as an ROS, the cells that use oxygen regularly will have this enzyme ready to go. And anytime there's a peroxide ROS formed, it can turn that peroxide into water and oxygen. So basically breaks this down into the harmless water and harmless oxygen gas. And that's the job of catalase. And peroxidase. So this is our sort of, you don't need to know anything here. I just wanted to show you that when you have a superoxide and you have this enzyme, the enzyme can turn it to hydrogen peroxide, and then you have another enzyme, the catalase, that converts it to water, right? So we have oxygen forming nasty product, another nasty product, and then ultimately, if you have these two enzymes, it can just form water. So we have a way of protecting our cells from these toxic forms of oxygen. When you are an anaerobe, right, like Clostridium, and you live your life without oxygen, there's no need to have superoxide dismutase or catalase or peroxidase in your cells. There's no reason why you would evolve those enzymes because you don't come across oxygen. And that is why oxygen can be toxic to an anaerobe, because when you give an anaerobe oxygen, it forms these nasty, toxic forms, and that ultimately kills the cell. This is something we'll do in lab. This is a catalase test to see if your cells have catalase or not. So all you're going to do really is put your bacteria onto the slide, and then you drop a little bit of hydrogen peroxide on your bacteria. And if your bacteria are able to break down that hydrogen peroxide, that means they have catalase. It'll look like this. It'll look like you have bubbles. Why are there bubbles? Because when you break down hydrogen peroxide, you make water and oxygen, and this oxygen is given off as a gas. So what you'll see in lab is that if a bacteria has catalase, it'll break down hydrogen peroxide into water, and then the gas will create the bubbling. If the bacteria does not have that enzyme, you will not have bubbling, and you won't have any reaction. Candle jars are special jars used for creating a high carbon dioxide content, and the bacteria that would like to grow and grow well at high, a little bit higher of a carbon dioxide content than the atmosphere are called capnophiles. So capno refers to carbon dioxide, and the candle jar is the way to go here. So what happens is when you light a candle and you seal the jar with your bacterial cultures, the candle will burn most of the oxygen away, and through combustion, right, we're going to make carbon dioxide gas. So the candle will use up the oxygen and then create more CO2 through combustion. And so this jar, right, will ideally have less oxygen, more carbon dioxide, which is a nice environment for our capnophiles. So that's called a candle jar. All right, let's take a break.